Good morning, guys. We are going over our chapter 3, section 5 notes here about cell division. It's going to talk about the process of mitosis and give just a brief overview of what happens. So, if you have your copy of the 3.5 notes, you can follow along and pause to fill those out with this video, or you can look at the PowerPoint slides as well that were in today's folder. So, cell division, think about when a pumpkin grows in size, and you see like during fall and hay rides and all of that stuff, you see how big pumpkins can get. They are increasing in the size of the cells as well as the number of cells. So this doesn't happen in just pumpkins. It actually happens in um, many of the other cells throughout your body. Not all of our cells do this and grow and divide, but there are quite a few that do. So the way we're thinking about it is with like little baby pigs and how they start out really little and then get really big. Or if you've ever had like a puppy or a kitten, you know how little they are and then they get huge. So the cell cycle is defined as the regular sequence of growth and division that cells undergo. So interphase is the first stage of the cell cycle, and during this stage, a copy of the DNA is made. It prepares to divide into two cells, and the cell's also getting bigger. Now, we will learn as we go through our notes, and then later this week, that interphase is actually the longest phase, and it probably takes about 90% of the cell cycle like time because it takes so long to copy that DNA and make sure it's copied correctly so that it can divide and be put into those two cells. So during this first stage interphase, the cell grows to its full size and then it starts to produce other structures that it needs. So it makes new ribosomes, it produces enzymes, it makes copies of the mitochondria and chloroplast. So that's going to help with those processes of photosynthesis and cellular respiration, depending what type of cell it is, either a plant cell or an animal cell. So it needs those specific organelles to help those functions as well. Then in interphase, there's an exact copy of the DNA made in the nucleus, and this is through the process of DNA replication. We'll probably look at this a little bit more later this week as well, but it's almost like taking apart a zipper and then putting it back together. So you have those two sides and then it builds a new zipper and then you get two zippers. So that is all during this interphase process. So replication is very important because each of those daughter cells or each of those two new cells at the end of mitosis have to have a complete set of DNA because that DNA carries all the genetic information that our cells need to be able to function and know how to process. So then once the DNA is replicated, cell division begins, and it's going to even produce more structures that help the cell divide into two cells. So the second part of the cell cycle is mitosis, and this is where the cell's nucleus divides into two, and through that process, one copy of DNA is distributed into each daughter cell. Now, mitosis is a lot faster of a process than interphase, but it still does take some time, and mitosis is divided into steps. So the four parts of mitosis, or the phases, are prophase, metaphase, anaphase, and telophase. So during prophase, the chromatin in the nucleus, so this is that genetic material, it's kind of just floating around in a little ball. This condenses to form double rod structures called chromosomes. And in our guided reading notes, we identified the structure of a chromosome and what it looked like. And then each identical rod, so each single rod in a chromosome is called a chromatid. And then like I said, in our guided reading, the diagram also showed the centromer, which holds those two chromatids together to create that chromosome structure. Then, as the cell works through prophase, it goes through the other phases as well, metaphase, anaphase, and telophase. And through this process, the chromatids or the chromosomes line up, the chromatids separate, the cell starts to stretch, and then new nuclei start to form, and eventually the cell starts to split and create two new cells. We will get into each of these phases a little bit more tomorrow. We're going to start a gizmo that's going to kind of walk you through the phases to show you what happens and give you a little bit more information as well. But the picture on this slide kind of gives you a brief summary so you can see how things move and stretch. Now, the final stage of the cell cycle is called cytokinesis. And in this process, this is where the cells finally separate and divide. So the cytoplasm divides, and it's going to form a new cell membrane if it's an animal cell. And then the organelles are distributed into each of the new cells, and they have about half of the organelles they need, but they each have a nucleus which has a complete set of the DNA in that nucleus as well. In a plant cell, we'll get to that, but that also kind of adds another component in this cytokinesis stage due to the cell wall. Okay, so like I had said, interphase is a very long part of the cell cycle, and it depends on what cell it is. So sometimes, like a human liver cell, completes the cell cycle in about 22 hours. So you can see how long it takes for the cell to grow, and then DNA replication takes a long time, and then it's preparing, and then mitosis and cytokinesis only take about an hour of that whole 22-hour process. So other cells might take less time, they might take more, and there's even some cells that don't divide. If you watched our cell cycle and puzzle, it talked about those like checkpoints, and some cells just kind of stay in a state of rest. So they grow and get bigger, but they don't ever divide. They just become like mature cells. So in animal cells, like I said, in cytokinesis, the cell membrane squeezes together around the middle of the cell, and that cytoplasm separates and pinches into the two cells. And then each daughter cell gets about half the organelles, and that kind of completes that process of the cell cycle and cell division for animal cells. But then in plant cells, they have a cell wall, so they have that more rigid structure. So this new structure called a cell plate forms, and it's used to like divide the cells, and that cell plate then creates the new cell membrane, and then through that process, a new cell wall is able to form and harden and give that plant cell more structure. So with DNA replication, this again is ensuring that each daughter cell or each of those two new cells have the exact genetic information that it needs to be able to carry out its activities. So in 1952, Rosalind Franklin was able to use an X-ray method to photograph what DNA molecules look like, and then her photograph helped James Watson and Francis Crick figure out the structure of DNA in 1953, so how it was actually set up. And to give you a little bit of perspective, 1952, 1953 happened less than 70 years ago. So, like, you might have grandparents that are in their 70s. So, this was happening during the time when they were born and growing up. So, understanding DNA can still be thought of as possibly like a relatively new idea, and understanding like how it's set up and how it's structured is pretty impressive. So DNA molecules look like twisted ladders, or some people say like a spiral staircase. They are considered to be a double helix, which means they have like two strands that are twisted together. Um, and then it's made up of sugar molecules called deoxyribose, which is where it gets part of its name. And then those sugar molecules connect with other molecules known as phosphates. And that deoxyribose and phosphate are what create the side or that initial structure of the ladder. Then each rung, or where you would like think about a step for the ladder, is made up of a pair of molecules called nitrogen bases. So there are two bases that pair up with each other, and there are four kinds of bases that make up DNA. And they are adenine, thymine, guanine, and cytosine. So we use the capital letters A, T, G, and C to represent them, so we don't have to like write out the whole name. And when we look at DNA structure a little bit later, we'll just use those letters. Now, each of those bases have to pair up with a specific base on the other side, and they can't like mix in with other bases, or it creates an issue with how the DNA is replicated. So adenine can only pair with thymine, and guanine can only pair with cytosine. So these pairings are key in understanding how DNA replication occurs. They have to match up with those other pairs in order for it to be completed correctly and function. So through the replication process, DNA begins when the two sides of the DNA molecule unwind and separate. So you can think about this as like a zipper unzipping, and then you have two sides of the zipper that are open. So then there are nitrogen bases, so those adenine, thymine, guanine, and cytosine. There's some of those pieces just kind of floating around in the nucleus, and they pair up with those open bases on each half of the DNA molecule. And then because of how those bases pair up with each other, the order in each new DNA molecule matches the order in the original DNA molecule. So once the new bases are attached, two new DNA molecules are formed. So thinking about the zipper, you unzip it, you open up those two sides, new little pieces connect to those open zippers, and then think about it as making two new identical zippers from that one original zipper. And if everything attached and formed correctly, you would have two working complete DNA molecules or two new zippers. So this is just kind of a representation of what DNA replication looks like. You have this initial chromosome, it has the DNA. As you can see, it starts to separate, and then there's called these DNA polymerase, and they kind of are where those um, nucleotides or those nitrogen bases start to go through and reconnect to create two new DNA strands. So once you have these notes done, please go back through the video if you need to fill in any blanks or if you missed anything. Otherwise, you're submitting your notes, and then you have your Edpuzzle today. Bye, guys.